Hello, everyone, and good morning. Welcome to the last of our webinars on the future of treatment in November, part of our Living Well with Prostate Cancer webinar series. It's my pleasure to welcome you today to today's session on artificial intelligence and machine learning, a new frontier against prostate cancer. My name is David James, and I am the head of patient projects at Prostate Cancer Research. So a little bit about prostate cancer research. We are a research-focused charity that's focused on the prevention, diagnosis, and treatment of advanced prostate cancer. By funding novel and innovative projects across world-leading institutions in the UK, we're working towards delivering breakthroughs in the prevention, diagnosis, and treatment of advanced prostate cancer, and ensuring better quality of life outcomes for patients. We're proud to fund research right across the UK at a number of different institutions, from Aberdeen, Cambridge, Cardiff, Glasgow, London, Newcastle, Norwich, and Oxford, focused on key areas such as bone metastases, radiotherapy, immunotherapy, and AI, with the shared aim of reducing morbidity, increasing survival rates, and improving quality of life for people with prostate cancer. Now, you'll be pleased to know I'm not going to be giving an in-depth update on one of our research projects today, as I have done in the last sessions, because I'm very excited that we're actually joined by a number of our speakers today who are actually leading on one of these projects. So I'm going to let them do the in-depth part themselves. So you actually get to hear from the people leading the project, not me. So some quick housekeeping for this session before we get started. As anyone who's joined us previously will know, all attendees' microphones and cameras are muted, and this is so that we can record the session and share it with folk afterwards, both people who are able to attend and those who weren't. If you have a question during the session, we will be opening up to questions at the end, but if you have a question at any point during the session, please feel free to drop it in the Q&A section at the bottom. You should have a sort of Q&A section at the bottom of your screen. There isn't a chat function available, but you can use the Q&A function for this purpose. So before I introduce you to our speakers, I just wanted to let everyone know about some of the remaining sessions we have in December and January. So these are our last 2 months in this webinar series. And so in December, we have 4 sessions on the theme of looking and feeling great. We have a session called Let's Talk Sex and Intimacy, brought to you in association with the Sexual Advice Association. Both patients and their partners are most welcome, and that's on December 1st at 1 to 2 p.m. Then on December 3rd, from 4 to 5 p.m., we have a special session by the same name, Let's Talk Sex and Intimacy, but for the LGBTQ community. And again, patients and their partners are most welcome to attend. Later on in December, we have 2 workshops, and these are brought to you in association with Look Good, Feel Better. And we have on December 10th, from 2 to 4 p.m., we have a skin fitness workshop. And on December 14th, from 10 a.m. to 12 p.m., we have a body confidence workshop. I hope anyone is, if they're interested in these sessions, that you do come along and attend. I think this is going to be really fantastic. And then in January, we have the final session of this series on the theme of supporting others. And that's brought to you in association with leading cancer charity, Maggie's. And this is a webinar which has been designed specifically for wives, husbands, and partners to help them understand and cope with prostate cancer diagnosis in the family. But patients are also most welcome to attend this. So this is taking place on January 10th, 2022. My goodness, we're almost there, from 11 a.m. to 12:30 p.m. And if you know anyone who you think might be interested in that session, please do pass those details on to them. So more information about all of these can be found on our website, the URL which is on the screen. That's www.pcr.org.uk/living-well. So now it's my great pleasure to introduce you to the speakers we have today. We have 4 speakers today, so we're beating last week's record of 3 speakers. We're joined by Professor David Wedge, who is the Professor of Cancer Genomics and Data Science at the University of Manchester. Dr. Daniel Woodcock, a Senior Data Scientist at the Nuffield Department of Surgical Sciences, and Professor Dan Brewer, Senior Lecturer in Medical Bioinformatics and Genomics at the Norwich Medical School. These are the 3 gentlemen who are leading on one of PCR's research projects. And they're also joined today by Dr. Antony Rix, who is the CEO of Lucida Medical, a company who's using AI and machine learning to make prostate cancer screening more accurate, accessible, cost-effective, and quick. And I'll leave him to tell you more about what they're doing in that space. So without further ado, it's my great pleasure to introduce you to our first speaker and hand over to him, Professor David Wedge. Over to you.
Thank you, David. If you just give me a moment, I'll just get my I'll share my screen. Okay, so in this the first part of this webinar, I hope to answer the question, what is AI? There are broadly two definitions of artificial intelligence. The first is as you see here, a computer performing tasks that would usually be considered to require human intelligence. And the second is a computer acting rationally in taking actions that maximize its chance of achieving its goals. The difference really between these two definitions is that in the second definition, there's some kind of sense or idea of agency of the computer actually exercising some degree of control over how it achieves its goals. So I can kind of illustrate this with a few examples. At one end of the spectrum, you have an example of a computer that plays chess. As you probably know, they can now make computers that play chess very, very well, play very, very strongly, can actually beat the strongest human opponents. But they are, these are not thinking machines. They're not exercising kind of conscious control over how they play chess. At the other end of the spectrum, we have something like Terminator, that is a robot that has essentially just one task, which is to terminate John Connor, but actually has exercises a lot of control over how it actually achieves that task. In the middle, we have something like self-drive cars. So self-drive cars are actually carrying out a number of different actions. So they have to carry out kind of route optimization. They have to avoid obstacles. They also communicate with the human occupant of the car. But they are not. They don't actually exercise control over really over how they achieve their goals. So self-drive cars are not yet acting rationally. They're not achieving the second definition of artificial intelligence. Another kind of distinction that's made between different types of AI is between what's called narrow AI and general AI. So narrow AI is type of artificial intelligence that is designed to carry out exactly one task. So examples would be virtual assistants such as Siri or Alexa, image or facial recognition software, email spam filters, or kind of marketing recommendations from companies such as Netflix. I also put into this category self-drive cars, because although they carry out a number of different tasks, actually they have one overall aim, which is basically to get you from A to B. On the other hand, general, or it's also called strong AI, is artificial intelligence that is able to solve problems across various areas, and basically all areas that a human can. Also included in the definition of general or strong AI is the idea that AI has mind and consciousness, whereas narrow AI can only act as if it had consciousness. So again, we kind of get back to this definition of whether the computer actually has kind of consciousness and awareness and control over what it's doing. The current day, strong AI doesn't exist, although there are a number of experts who think that it may come about within the next few decades. From now on, I'm going to be talking about kind of real AI as it exists today, and I would say all of the categories of all of the AI that exists today falls into the categorization as narrow AI. Although we call it narrow AI, actually AI is used across a vast range of fields. generally, this kind of covers the goals of AI. So generally, they fall into these categories: problem solving and reasoning. The example shown here is at the top of a computer that can basically solve the Rubik's Cube. Knowledge representation, which would be about creating some representation, often a hierarchy of objects or events, as you see in the second figure. Planning, which can be again, something like getting from A to B, like a self-drive car. Natural language processing, which is all about extracting meaning from either spoken or written language. Perception, which is things like image recognition. Motion, which is kind of what's associated with like robots that are really quite widely used, for example, in manufacturing, like car manufacturing and that kind of thing. And finally, learning. So the rest of my talk is actually going to be about learning and specifically machine learning. So what is machine learning? So machine learning can be considered to be a class of computer algorithms that improve automatically through experience with data. So this is a kind of a cartoon illustrating what I mean by that. So with any machine learning technique, we have a set of input data, and we want the machine learning algorithm to make predictions on that data. So a simple example would be that we might have images of 1,000 cats and images of 1,000 dogs, and we want the machine learning algorithm to tell us which images are pictures of dogs and which ones are pictures of cats. The way that most machine learning algorithms work is that they start with basically random predictions. Then we present the data to the algorithm, and the algorithm is tweaked to improve the predictions. And if we do that with enough examples of pictures of cats and dogs, then the algorithm will learn to make better predictions. So they can then, when you present the algorithm with a new picture of a cat or a dog, it's able to accurately classify that as the right animal. So there are each machine learning method has what we call a model, which is the kind of the structure of the data within the machine learning algorithm. Sorry, I'll just go back. These can be broadly classified as 5 different models. The first is neural networks, which are modeled on the human brain. So they're made up of a number of small processing units that we call neurons, which will take an input. They'll kind of convert that. They'll do a small computation on that input and output a different value. That value will then be passed on to another neuron, which again does a bit more computation and then passes on the output to the next neuron. So you can see that a neural network on the top right of the screen. Neural networks are normally constructed in layers, with each layer of neurons passing on an input as an output to the next layer before making the final prediction by the output layer. The other methods that I show you here are evolutionary computation, which are based on kind of the genetic code, and which we each pass on to our children. So with genetic algorithms or evolutionary computation, we actually have several different algorithms which make a computation and make a prediction. Again, using our example, they might predict whether you've got an image of or a cat or a dog. But because we have multiple different genetic algorithms, we can then subject them to a process of evolution. So we can make small tweaks to the algorithms, which we would call mutations. We can also take bits of one algorithm and combine them with bits of another algorithm in a process that we call crossover, which is basically the kind of computer equivalent of sex. So after a number of iterations, you kind of evolve the population with the aim of improving the offspring, so that you get really good algorithms that make good predictions on the data. the other methods that I show here are particle swarm optimization, which is similar to genetic algorithms in that you have a whole population of different solutions, and they act a bit like a swarm of bees or a flock of birds, and they kind of move around the kind of space of possible algorithms to try and search out and find the best algorithm for really solving the problem. And the final two methods that I show you here are based on networks, either decision trees or Bayesian networks, which give you more kind of interpretable information to tell you how the algorithm has actually made the decision that it has. So I want to say a bit about one particular method, which is really being very widely used and has received a lot of interest in recent years. And this is deep learning. So deep learning is actually a type of neural network, but it's called deep learning because with deep learning networks, you have a really large number of different layers. So I showed you earlier the layers of neurons, and with deep learning, we have a really large number of layers. So you may have kind of tens of layers, each of which take an input and then pass on the input to the next layer. The reason that deep learning has multiple layers is that they can actually learn features of data. So deep learning has been quite widely applied to image analysis. And what it does is you can see in the bottom figure, what it does is it will take the raw data, which will just be individual pixels within an image. It will then identify kind of local features of the data, but allowing for the fact that they may be kind of rotated around, or they may be larger in some regions or smaller in other regions, and then they actually match them to or group them together. So you can see that in the example here, we've grouped together lots of images of eyes and noses and different kind of features, and then it puts it all together to make a whole face. So it's basically starting at the really fine level, the pixel level, and then building up the image one step at a time into the whole face. With machine learning, as I mentioned earlier, we often start with a kind of a random solution, but then we train that algorithm to improve the algorithm. There are different methods that can be used for training. The one that I mentioned earlier, where you have images of cats and dogs, would be considered as supervised learning. So then we actually know what the correct classification is, and we can train the algorithm over several iterations to improve the classification so that it correctly classifies cats as cats and dogs as dogs. The second method is unsupervised. So this can be used when you don't actually know what the classification is. So you might have lots of pictures of animals, but you don't actually know what animals they are. And you just want the algorithm to discover for itself what the different types of animals are. So in that case, you would use unsupervised learning. And the difference between these two can be considered that the first one is carrying out classification, whereas the second method, the unsupervised learning, is carrying out clustering. The third method is called reinforcement learning. This can be applied in much more complex situations. So for example, if you have a computer and you want it to play a computer game, then what you can do is just get it to play the game hundreds and hundreds of times, and then it will get some kind of feedback, because it'll know whether it won that game or whether it got a high score on that game or it got a poor score on that game. If it gets a high score, it will then, the next time it plays the game, it will try and do more of what it was doing the first time. If it doesn't get a very good score, then it will do less of what it was doing the previous time. So often machine learning methods are described as black box methods. What we mean by that is that we provide an input to the machine learning method, and it gives an output, but sometimes we don't know what's gone in the processing in between. And that's because the machine learning algorithms often use very complex methods. They're taking lots of different data, they're doing lots of computation on it, and then just spitting out an output. This is fine in a lot of situations. People are quite happy with that. Just if they know that an algorithm is working well, they may not really mind not knowing why it's working like this. But increasingly, machine learning is being used in in quite sensitive situations, for example, working with customers where you may be, the computer may be making a decision whether to give somebody a mortgage. In that situation, it's really important that we know, why the AI is making the decision it is. So there has been a move in recent years towards explainable AI, and what this does commonly is it looks at the inputs and the outputs, and it looks at the processing that goes on in between, and identifies, well, what is the pathway from the input to the output. And so from looking at the kind of pathway through the model, can we see, well, what are the inputs which have most effect on the output? So what are the factors that have been included in the calculation that mean that somebody has been declined a mortgage? And increasingly, there are methods used in AI that do allow us to extract that information from the black box and kind of open up the black box and find out what's going on inside. So in my final slide, I just kind of list some of the applications of AI, but basically AI is being used in just about every field that involve humans. So I won't kind of go through this whole list, but I'm going to pass on to Dan Woodcock, who's going to tell you a bit more about the first of these areas, how AI is used in healthcare.
Hi everyone, I'm Dan Woodcock, and as David said, I'm a senior data scientist at the University of Oxford. I'm here to talk today about AI in healthcare, and essentially why we're doing what we're doing and why we're doing it now. Can everyone see my screen okay? I assume so. Right, let me know if you can't. Okay, so AI in healthcare. So why now, really, is the question. So we're at a sort of unique juncture at the moment, really, in that we've got sort of three things that sort of have aligned very well, and we want to exploit those. So the first of those is big data. So nowadays, with the rise of computers and so on, we've got a lot more data and that's available to us than we used to. The second, we've got powerful computers. So you've got the sort of actual sort of raw power to process that data. If you try to do what we're trying to big data analysis on like Commodore 64 or something, you'd have problems. And finally, we've got advanced algorithms. So there's no point in having the data or the computers if you don't have the algorithms to sort of process that data on those powerful computers. And so really, it's all of these three things together that allow us to sort of approach things like healthcare improvements and other sort of fields that David mentioned. Healthcare improvements are particularly of interest because obviously we've got sort of more centralized data servers and so on, and biobanks and things like that. So that's really what's bringing this sort of big data side of it together. In terms of how we might use AI in healthcare, I've given some examples on the right here. So we've got things like natural language processing. So that's a type of AI where it does what it says, really. So for example, so after a clinic, a doctor might sort of record their notes and then put them into, like traditionally, they'll be sent off somewhere to a secretary or to a sort of third-party sort of station, I guess. And then they'd be transcribed then, and then that's what would then be sent to GPs and so on. But obviously that's laborious, it takes doctors' time, it takes everyone's time. So natural language processing techniques, you can just record it down and then that will be transposed by a computer and then sent to the system. Another example of AI in healthcare is robotic assistance in surgery. So, yeah, obviously a robot, say, yeah, if you're doing a prostatectomy or something, or yeah, that's got a lot of sensors on the outside of it, not just the things that feed into the human, and they can actually sort of automatically adjust sort of things like pressure and orientation, stuff like that, without the human explicitly having to do that. So, and that will give sort of better outcomes and sort of more precise surgical margins and so on. Another thing is remote monitoring, so alerts. That would be, so say if you're wearing an ECG holster, if you can predict if you're about to go into a heart attack or an atrial fibrillation episode, then that would be obviously very good for you, and they can send that information into relevant authorities, or even, you know, dining an ambulance for you if necessary. Automation, yeah, automatically send samples to the right place and automate the sort of yeah, the processes that are involved in after surgeries and so on like that. And then finally, we've got machine learning, which we've spoken about already, David has, and I'll go into a bit more about that myself now. So David explained what it is, so I won't go into that again, but I think the way I'm sort of conceptualizing it here is that it's teaching the computer to recognize patterns in the data, much the same way as the computer does. So we can recognize patterns that can inform things like risk profiling, and so on. And then so on the left here, I've sort of split the sort of healthcare domain into three loose sort of areas, really. The first one is risk profiling and disease prevention. So that's, prevention, I think, is the operative word here. And with machine learning, you can sort of investigate things like social demographic factors or lifestyle factors, and then sort of coalesce those into sort of a recommendations or something like that. So, lifestyle, obviously, stop smoking and things like that. So obviously, that's very much focused on prevention. The second, clinical, is more focus on the cure. So you can use machine learning methods to improve detection diagnosis. So you can't treat unless you've been diagnosed. And then once you have been diagnosed, then sort of the clinical decision support. So as David was saying, with regards to black box, so a clinician needs to know why they're making a decision. And so rather than telling a doctor, okay, you should treat this patient, or you shouldn't, you could just present the information to them as why you should treat this patient and why you shouldn't, and that would feed into whole clinical decision-making pipeline. And finally, in this example, then we've got targeting treatment. So we can, not all treatments are suitable for all sufferers of the same disease. So classic one, which I think we'll talk about later, is HER2 positive breast cancer. So the things can get more complicated than those things, and so we want to sort of be able to think of a framework where we can actually target treatments effectively to those who need them most. And finally, we've got research, which sort of feeds into both prevention and cure. And that's scientific discovery. So that's really understanding what the disease is, what processes involved, and then feeding that into ultimately drug development and other sort of treatment outcomes that we want to promote. Okay, so I've got a few more details on some examples of how we can use machine learning in healthcare. The first of these is electronic health records. So as I mentioned briefly previously, data from patients is now routinely stored electronically, and that is pretty much everything. So your date of birth, immunization record, whether you went to hospital with a broken leg when you're 12, literally everything is going to be stored, especially moving on to the future. And so these undoubtedly contain a lot of information that is relevant to disease. So it may be that when you broke your leg when you're 12, they found something else on the x-ray that then may be linked to a disease that you'll get in 50 years' time. We don't know. It's so much information. It's just mind-boggling to think about how to search it. So really, this is looking for a needle in the haystack, hence this image on the right. But we believe that AI can actually be used as a tool to find those needles in the haystack. So like I say, it's sort of like recognizing patterns. So what the AI would really want to be doing is extracting those really small patterns and relationships, and then sort of bringing them to prominence. So then you can sort of research them further and use them. So this is a sort of depiction, like a sort of characterization of what electronic health records would be like. So in now, so that is where the patient is in their life. And so they've got the previous health records, and then you've got things that will ultimately happen to you in the future. So you want to essentially use what's happened in the past to predict what is going to happen to you in the future. And then sort of forewarned is forearmed. And so then clinicians can sort of use that information to then sort of, either step up screening protocols or determine risk factors, or telling you to stop smoking because it's really important now, and things like that. And roughly, this is broadly a way a computer would do it, is that you'd input all the information that up until now, obviously, into a vector, which is a mathematician's sort of word for just an array of numbers. And you put that sort of in a way the computer can understand, so demographics, diagnosis, medication, and that would always get fed into a neural network or some other machine learning algorithm, then that would be used to form a prediction. And in this case, it's between heart failure, and assigns a probability of patient one getting, I've got a 20% chance of heart failure, and so on, and the probability of admission to hospital in the next year, and things like that. And you can imagine that you could obviously expand that to lots of things. So yeah, seemingly irrelevant bits of data or information like your height when you were 12. Could that be indicative of, excessive, a lot of testosterone, which then could be indicative of whether you might get testicular cancer later in your life? So there could be potentially really, really minute details that you wouldn't ever think of normally, but that's actually important in sort of prediction. A second big area of machine learning research and in healthcare is image analysis. And so I guess this is probably one of the biggest success story of machine learning in sort of modern times, really. And these predominantly rely on the deep learning methods that David was talking about. And as he said, it's been successfully used in many fields, from military, farming, transport, driverless car, manufacturing, airport security, medicine, and lots and lots of other things. And so on the right here, we've got essentially how a driverless car might sort of segment up its current image of a road and then identify what's going on there, what they are, and any potential hazards that might occur. And then they can use that information further on to then feed into their sort of response to these potential hazards. Yeah, I was talking about how imaging can be used in healthcare. So we've got radiology, MRI and CTs, and pathology, so histopathological grading. So in this case, I've used the example of a Gleason score, and this is really to augment the pathologist's own sort of decision, just lead them to help them make their decision, but not actually make their decision for them. So this is a bit like what David was talking about, opening the black box and just really helping people to understand why they're making their decision, not to actually enforce that decision on them. And you can see from this example, then identifying areas of sort of the various different Gleason grades, Gleason 3, 4, and 5. And pathologists can really zoom in on these areas and then confirm that that is indeed those actual Gleason grades, and then use that to inform the diagnostics for and grading for the patients. If I now put augmented reality in this surgery, in cancer care, so yeah, people are doing research on things like having a PSMA PET, which is a sort of marker for prostate cells. And so if that's injected, a sort of marker for that is injected into the patient while the surgery is being performed, then the surgeon can use, like, sort of see the operation through their normal robot vision, but then that will be augmented with the PSMA PET information, which the AI will automatically sort of superimpose onto that image. That will show them exactly where the cancer is, and that they can cut it out more effectively. Okay, ultimately, I've used Gleason as an example here, but things like image analysis and also further wider field, we want to look beyond Gleason, beyond these sort of just augmenting, and actually sort of find features that are indicative of poor outcomes. So in the case of Gleason, we sort of get biopsies, and that will get sent to the histopathologist to perform the Gleason grading, and then we'll use AI to essentially augment that process, and that will be used to determine the severity. What we would like to do is use that outcome, so the poor outcome in this case, in this bottom schematic on the left, and use that to train the network directly. So then the AI, the machine learning algorithm, would learn features itself that are not directly linked to Gleason that would be indicative of poor outcomes. So we're really cutting out the middleman there. So Gleason essentially is a proxy for severity. We can maybe be able to accurately identify that, but it would only be as good as a Gleason grade is. Whereas if we can actually identify new features, which some of these are sort of indicated on the right, then we can actually sort of train the algorithm to point pathologists in the direction of these new features, which actually might be more predictive of poor outcomes. And then finally, I want to give an example of personalized medicine with AI. So standard sort of, I guess, approaches currently use what's called population-based approaches. So you pull everyone together and then form predictions based on simple rules. So on the right here, we've got simple disease severity and blood pressure graph. So you think the higher your blood pressure, the worse the disease or whatever. And Mr. Smith has been put to the left side of this rule line, which is also called a decision boundary, which is what a neural network calculates when it wants to make a decision. And then, so Mr. Smith would be assigned that he's in the, well, in this case, the red category. Obviously, that's pulling everyone together like this doesn't always work. So the situation is far more complex in reality. And what we should actually be doing is tailoring our predictions towards the individual rather than sort of segment them as part of a population. And so this is what is really called personalized medicine. And it's a sort of very deep field, very complex, not only scratch the surface here, but essentially it's what we want to do is stratify patients based on things like the subtype, their risk, their prognosis, and their treatment response. So we've got an example below from Michaela van der Schaar's group at Cambridge, and she's created a personalized medicine classifier for breast cancer. And so if you had these options of treatment, here at the top, no treatment, radiotherapy, chemo, and chemo plus radiotherapy, and you were to perform a population-based analysis, then you see that no treatment, it gives you the worst chance of recurrence, which is the scale on the left here. And then radiotherapy is the second best, and ultimately chemo plus radiotherapy would be the best at a population level. But when you look at it from an individual patient's perspective, and you can feed in all the information about that patient, then the risk sort of profile could look very different. And in this case, you see that the machine learning algorithm predicts that if you did no treatment, you would drop to 35, and then the profile would essentially completely change. So that chemotherapy on its own would be the best option, presumably because the patient has got some adverse reaction to radiotherapy or something like that. But this is really sort of how we would like the future medicine to sort of progress towards, especially in what I do. Okay, so to round up then, there's been a number of successes in AI applied to healthcare, such as drug discovery. So AlphaFold, which is made by Google DeepMind, that made the news because it sort of was able to solve one of the grand challenges, I guess, which is predicting the protein structure from its amino acid sequence, which is incredibly useful when you're trying to invent new drugs. We've got improved detection of cancer through various means, some which we'll talk about later, I think, and more accurate surgery, like robot assistants, which I'll briefly touched on, and then the monitoring, so the prediction of adverse events. So if you want to be screening frequencies, what tests you should be doing, and so on. So these are things that we've got under our belts really, and have proven that they can work. And so I just wanted to end basically. So we've got this sort of magisterium of AI that is out there, but very little of it is actually translated to the clinic, and basically why that is. And so that's the challenge, one of the challenges of AI in healthcare at the moment. And so the first one, I think, is trust. So again, we've spoken about the black box before. So how does the patient and the clinician trust what the computer is telling them? As you see with my internet connection, it can't even sort of stay stable for that long. So how would, can we sort of improve the trust in the patient experience really, and just everyone's confident that the patient's getting the best care that they can? And ultimately, the first start to that is unlocking this black box. So a lot of our work is sort of understanding why the machine learning algorithm is making the prediction that it does. Obviously, you've got other concerns with patient confidentiality and things like that. So if you're a patient, you don't want your data going off to everyone, so they can train our algorithms, whether it's anonymized or not. And so there's issues there that obviously need to be discussed. Data availability. So algorithms are only as good as the data they're trained on. And so what you find in lots of sort of Western countries, I guess, is that things like Black people are underrepresented in the data like pools that we've got out there, and then obviously the machine algorithms are then tailored towards white people or people from other ethnicities, and that's obviously not very desirable, to say the least. So we need to address that basically, and then obviously when you've got a algorithm that's been trained on some data that might be publicly owned like NHS, but who actually owns the algorithm and things like that. So we've got things like the bureaucracy all involved in that, which is in IP and all these things. So although AI in healthcare has shown a lot of promise, but there's still some challenges to overcome. And that's the end for me. Thank you.
Okay, so I think it's over to me now. Thanks very much, David, for inviting us to speak today. Just get my slides up. Okay, so I'm hoping that you can see those okay now. Great. So So David started off by giving us a broad introduction to AI and machine learning, and then Dan focused down onto the aspects in healthcare. But I'm going to be even more specific and talk about a project that we've been funded by Prostate Cancer Research in the area of prostate cancer. And this is looking at the measurements we can take from cancer cells and how we can use that to divide up patients into different types of prostate cancer. Okay, so let's start off by talking about the prostate. Many of you will know this already, but it's a good baseline to start with. The prostate is a small gland that's part of the male reproductive system. It's about the shape and size of a walnut, and it's located below the bladder. It surrounds the urethra, and its main function is to release a fluid component of the semen. And prostate cancer is malignant growth within the prostate that is started in prostate cells. Now, prostate cancer is more commonly a disease that men die with rather than of. So most men we know, when they die, will have some detectable level of prostate cancer, whether they've been diagnosed for this in the clinic or not. In studies of patients that have died from other means, like in car accidents, we know that in over 50% of patients that are over the age of 60 will have some detectable levels of prostate. Now, this hasn't caused them any problem at all. They don't know it's there, and they've had no treatment for it. But it does exist. So we're not sure why this happens, but we think that the prostate is an inherently abnormal organ. And so over time, it will develop tumors. Now, speaking about this is all about prostate cancer that hasn't been detected. But let's talk a bit more about prostate cancer that is detected by the doctor. We know this is a huge problem in the UK and elsewhere. It's the most common male cancer by far, and it affects about 1 in 8 men overall. We know the incidence of prostate cancer increases rapidly with age, and there's other risk factors like ethnicity and family history. So let's have a look at those men that are diagnosed with prostate cancer in a bit more detail. Now, the good news is that although a massive number of men get prostate cancer, over 90% of them will survive for greater than 10 years. And that's really good news. So if we look at this bar here, this represents all the men that have been diagnosed by the doctor to have prostate cancer. Now, roughly half of those men will be given some sort of treatment. So this half will be placed on active surveillance, and they will be reviewed regularly. But this half will have some sort of treatment like radiotherapy or prostatectomy. Now, this little orange bar here represents men who have actually died from prostate cancer. Now, you might think, oh, okay, this is very small. It's not something to be concerned with, but actually it's a huge number of men. It actually represents over 10,000 men dying every single year in this country. So that is a massive concern. How do we focus on improving the outcomes for these men? The other thing that we need to be concerned about is that we've got a huge number of men being treated, and these have life-changing side effects. So we want to make sure that people are getting the correct treatment to avoid, so that they're not having unnecessary side effects. So the picture I'm trying to paint to you is that prostate cancer is complex. It can be divided into 2 categories. We've got the pussycats, where the cancer is slow growing and just sits in the prostate. It probably is not going to do you much harm. But we've got the nasty ones, the tigers, that are really aggressive and spread and will lead to to death if not treated correctly. So where are we clinically? We're really good at the extremes. So if you've got a pussycat, then we know what to do. If you've got a really aggressive prostate cancer, we know how to treat you. But we've got this middle region where we're not actually that sure what to do. And that means, to be on the safe side, we treat everyone. And so everyone gets the side effects, although they may not be necessary. So the big problem that we examine is, how do we distinguish the tigers from the pussycats? Now, one possible answer to this problem would be to identify subtypes of prostate cancer that are defined by the biology in the cancer cells. And that's really what our project is about. Subtypes have already been mentioned in the context of personal medicine. We really want to be able to say exactly what the disease is. And the reason we want to do this is so that we can make sure that the patients that have that type of disease are getting the correct treatment. And encouragingly, it can also help develop targeted drugs. Now, we've got a good example for this in breast cancer. So in breast cancer, there's 5 major agreed subtypes, and these are based on 4 markers that can be measured in the cancer cell. Different types lead to different treatment pathways. And if you look at the statistics, you see that after 2000, when these subtypes were introduced into the clinic, you get a massive drop-off from drop-off in females dying from this disease. Now, subtypes is only one part of it, but it is definitely a part that has helped amazingly so. The other thing to mention is Herceptin, which is one of the drugs that was developed when the biomarkers were discovered. So one of these markers, a drug has been developed specifically to interact with that, and that means for that type of prostate cancer, which is called HER2, this drug works amazingly well. Okay, so that's the problem. I'm now going to quickly talk about a bit of cancer biology. So what's going on in the cells that have been turned into cancer cells. So the place to start is DNA. So DNA is found in every single cell, and it's the instructions that make up that describe how to make the functional units of the cell. And these functional units are called proteins. Now, DNA is a long molecule that's made up of 4 different letters, A, C, G, and T. And we find that the DNA is responsible for what makes us human beings different from each other. So there's about 0.1 to 0.4% of differences, of changes in these letters between us, and the total length of DNA we find in each cell is 3 billion letters long. Now, a good analogy for the genome is a cookbook. So the cookbook is the DNA with all the instructions on how to make cakes, and each recipe is a gene and makes a different functional unit of protein. Now, the reason we're particularly interested in the genome is that cancer is a disease driven by changes in the genome. Okay, so the DNA sequence in cancer cells are different from those in normal cells. So it'd be really great if we could measure the genome, read what it says, sequence it in tumors, so we can work out what changes have happened in cancer, so that we can understand how it develops and how we can treat it. Now, the first length of DNA from a human was released in 2003, and it took 13 years to do, and it cost probably £2 billion. So if it kept at that rate, it would be impractical to do the research that we do. But fortunately, the price has dropped massively, and now it's probably around about £800 per tumor sample to sequence the DNA. And this is largely down to an amazing new bit of technology called next generation sequencing. I'm not going to go into detail on this, but Illumina are the leading company doing this. And the basic idea is to take a length of DNA, break it down into small chunks of roughly 150 letters long, and then generate multiple copies of these bits, and then measure each of these little groups of little bits. Now, this is great, but the problem is that we then have to basically perform a process called alignment, where we've got a jigsaw puzzle where we're trying to get all these bits and align them together to get our final sequence. Now, this is possible, but it really requires some high performance computing to do it, i.e. supercomputers. But now this is a regular thing in research, and it's becoming increasingly common to do this in the clinic as well, to read the sequence of the tumor cells. So what type of changes do we get in the cancer genome? We can see here, so the top line is what the sequence would be in a normal cell, and then you can see below what it occurs in the cancer. So you get things like substitutions, there's letters, deletions, insertions, changes in the numbers of areas, and also even you can get breakpoints where the DNA gets shuffled up and rearranged. Now, why is this important? Because it changes what functional units are produced. Okay. And it helps us decipher the mistakes that lead to cancer. Now, to help go from a change in an individual to whether this actually is driving the cancer, prostate cancer, we need lots of patients so we can see what similarities there are between those patients. Now, and that's the sort of like first layer of data that we look at, and it's pretty huge. Okay, the second layer of data is RNA. Now, before, when I was talking about the cookbook, with the recipes being the genes and the result being a cake, for example, or in this case, a functional unit of protein, I actually missed out an intermediary step in this process, and that's a molecule called RNA. So basically you get DNA transcribed into RNA, and then it's used to create the protein. So in our cookbook example, this would be like the chef writing out the recipe. Now, why would you want to do this? You want to do this so that if you want to produce a lot of a particular protein at any time, you can have you create a lot of copies of the RNA and then create them in parallel. Okay. This means if we can measure the number of copies we have of a particular RNA, we get an idea of whether the cell is trying to use a lot of that protein or not. In other words, how much a gene is turned on. In cancer, we find that a lot more of certain proteins are produced compared to normal cells. These are the proteins that help the cancer grow, and certain proteins are not produced at all, those that would block the cancer. So this is the second level of data that we look at. The third and final level of data that we look at is methylation. Now, DNA methylation is the main way that the gene activity is adjusted, i.e. methylation controls how much RNA is produced and hence how much protein is produced. And it does this by putting some chemical groups on the DNA. This is an example here. Like if it's methylated here, then basically this prevents the cell from reading this part of the DNA. So it sort of like blocks that DNA being read, and so the function units for that area cannot be produced. In our cookbook analogy, methylation is the equivalent of putting a paperclip over 2 pages so you cannot read the recipe that's in between. Okay, so we've got this huge amount of data, and they're all measured by the sequencing technologies that I introduced earlier. So it's a massive amount of data, And we really need machine learning to help us find the patterns in that data to work out what's going on in the cancer cell. So I'm quickly going to introduce you to two machine learning approaches that we've been working on. One is the Tiger test, and that works just on data at the RNA level. And the second is EVA Types, that works at the DNA level. Now, the important part of both these approaches is that they both use methods that's interpretable. We've mentioned the sort of like black box approach before. Now, we're trying to move away from that because it's important that we know what genes or proteins are contributing to a particular subtype. So, because this will help us gain a greater understanding of the biology and hence what is making that type of prostate cancer the way it is. This will lead to better treatments for those subtypes. Okay, so let's talk about the Tiger test or doesnt. When you're diagnosed with prostate cancer, you have a biopsy taken. This is when a small amount of tissue is taken and it's looked under the microscope, and it's that determines whether you have are diagnosed with prostate cancer or not. Now, to measure the RNA, we need to mash all of this tissue together, extract the RNA, and then measure it. Now, this mashing of the material has an effect on what we can do with the RNA. Now, if a sample is dominated by only a handful of genes, then it's quite easy to spot the different types of cancer. So this is an example in breast cancer. So in this sample, the cells are all dominated by this red signal from genes. So when we mash it all up, it's still red. When this yellow one's mashed up, slightly orange, but it's still yellow really. And the green, well, the dominant green goes to the green. This means that sort of like if we look at a few rounds of this, a reasonable set of patients, and ask the computer to see if they can spot the different types, then it's fairly easy to do. But prostate cancer is different. It's hugely complex disease with no clear over or underrepresentation of genes. So what basically happens is when we mash it all together, we get just a brown blur mess. Okay, and when we try to apply the same methods that were used in breast cancer, we can't do it. It just doesn't work. Okay, this is in part due to the math used for these tests, where a sample can only ever be part of one group. It can never be in multiple places at once. So we've applied a bit of clever maths called latent process decomposition. And what this does is help reverse the mashing up process by picking up on patterns that are common across hundreds of samples and then reversing it so that we can work out what genes are turned on and off in what proportions in what samples. In prostate cancer, we found a characteristic pattern that displayed particular poor outcomes in patients, and we call that doesnt. These are patients with Tiger cancers, and so they can be targeted for treatment. So we believe that Tiger test can be used, could save thousands of men unnecessary treatments and its side effects. So the second AI approach that I'm briefly going to look at is EVA Types. Now, this is based on a probabilistic neural network architecture. And one of the key modifications is that it retains the ability to associate the inputs with the features and the outputs. So in this model, we took DNA changes, so not RNA this time, but DNA at the moment. We've developed this neural network that also takes into account how the cancer has progressed, and it gives us two outputs. Firstly, what are the important changes in the DNA? And secondly, a classification into two groups. I'm just going to flick this up here to show you a bit more of the detail, but I'm not going to go into it here. And the results were that patients classified as one particular EVA type had a shorter time until the cancer would come back. And this was validated by 2 independent approaches using 2 different data types. So this is a really solid finding that we'll be able to classify cancers into 2 different types. But one of the really interesting findings about this is that it's not only the differences that occur in the DNA that are important, but it's when these differences occur. Okay, the ordering of the changes in the DNA matters as well. So, for example, if you had a change in a gene early on, then the cancer would develop in one way and the sample would be a particular subtype. But if it occurred later, then it might not alter the path of that cancer at all from the other subtype. Okay, and so this finally brings us on to the prostate cancer research project that we're really happy that they've agreed to fund. So we've got access to an amazing collaboration called the Pan Prostate Cancer Group, which is a collection of the major groups in the world that are working on this type of data. We've got a data set that has over 2,000 donors. It's got DNA with whole genome read from the cancer samples and from blood samples. It's got RNA and it's got methylation. And importantly, it's got really good clinical information and scanned tissue images. So we've got this amazing data set. We've got myself, David Wedge and Dan Woodcock from Manchester and the University of Oxford working on this, and it's going well so far. So what we're doing, we're basically taking this data set, we're applying doesnt to it, we're applying EVA types to it, but we're also extending these approaches so that they will work with multiple levels of data. Now, we're not just looking at the approaches that we're interested in previously, but we're also looking at all the other subtypes that have been reported in the literature by other scientists. And we've got 2 goals here. One is to look at whether we can produce a classification framework for primary subtypes, and by primary I mean from the cancer when it's in the prostate and confined there. And we're also interested in looking at whether there's subtypes in the metastatic tissue, i.e. when the cancer's spread to other organs, can we work out what is occurring there? And the whole aim of this is to get a better classification framework for work in the clinic using these biological markers. So thank you for listening. I don't think we're doing questions now, but at the end. So I'm going to stop sharing there and pass you over to Dr. Rix.
Dr. Rix, you're on mute at the moment.
Brilliant, thank you. And can you see my screen now?
Yep.
Super. So I'd now like to talk about some work that we're doing at Lucida Medical, which is much closer to the clinic than the previous presentations. In some ways, it's a bit simpler, but we're hoping to make a very significant difference to prostate cancer patients over the immediate next few years. The things that bring us together at Lucida Medical are really a number of challenges, and the previous speakers have alluded to some of these. The thing that makes me most passionate about is late diagnosis of cancer. Although 90% of prostate cancers don't necessarily progress, the issue is we're picking up that dangerous 10% rather late. The other side of the coin is, at the moment in prostate cancer diagnosis, a lot more patients are, with those pussycats are diagnosed than we would really like. Many of them go on to receive treatment that's actually probably unnecessary. The biopsy procedures alone are very expensive and quite painful, and if we can save those, we've got something good. When men are diagnosed with the lowest grades of prostate cancer, increasingly they're referred to what we call active surveillance, and that will and that itself, there the disease is monitored. over time, but it's quite a complex and expensive process. We want to make treatment better so that fewer men who are treated actually have significant side effects. And of course, in the NHS, we've got issues of resourcing and waiting times and very significant variability in the health that can be offered. These are the issues that brought us together. And 3 years ago, I started working with Professor Evis Saller at the University of Cambridge. I'm a serial entrepreneur with a PhD in machine learning. My first startup, which was based effectively on machine learning and AI 20 years ago, was bought by one of its customers 10 years ago. We didn't even call it artificial intelligence or neural networks then because they were quite unfashionable. Professor Saller runs a large research group at the university here, and she's a recognized pioneer in the application of medical imaging to cancer in general, and now focuses on machine learning applications. Lucida Medical is a startup company. We received funding earlier this year, led by a London-based financial institution called XTX Ventures, together with an American investor called Sawson Ventures, the University of Cambridge, and Prostate Cancer Research. We're developing software that's really focused on helping make radiologists as accurate as possible when they go through a critical new step of finding cancer using medical imaging. And I'll explain where the benefit of this is in a moment. Our software is called PI, or Prostate Intelligence, and it outputs a number of things. It helps identify targets, score risks, produces reports, and produces volumes and segmentations. We're actually applying a range of the artificial intelligence techniques that were described previously. I personally draw the definition of artificial intelligence quite widely. It's about teaching computers to do things that conventional programming languages can't do. So we make use of, for example, machine learning-like methods in image processing. But we apply the deep learning algorithms that were introduced earlier to identify what is the prostate in an image. We also identify where suspicious areas are, and then we use machine learning methods to estimate risk. But this is a good example of artificial intelligence methods in practice. And where we fit in, a patient firstly will have the blood test, the PSA test, and an MRI as normal. MRI was introduced specifically to help avoid biopsies, and one of our challenges in the UK and around the world is that it's doing that, but it's only getting the numbers down a relatively small amount. Our software helps the radiologist by plugging in to the workflow, the workstations that they use to identify which patients don't actually have any suspicious areas, so they can rule those patients out, or to locate cancers, as you can see from this highlight. It's a CE marked medical device, which means that it's already available in the clinic, and we're currently working on pilots. And the result of using PI is that the whole clinical team, as well as having the raw radiology images, now have these annotated heat maps so they can see where lesions are. They've got volumes so that they can much more accurately quantify the disease and perform biopsies and manage treatment. And ultimately, it's all about making sure that the patient gets reassurance if they don't have disease and exactly the treatment needed. And our take on the tigers versus pussycats discussion is that actually a large proportion of the pussycats will never develop these low-risk cancers. And so the clinicians we work with term them insignificant cancers. I'll mention them in a moment. And really what we're trying to do is have fewer biopsies. That actually should reduce the numbers of pussycats, insignificant cancers that are diagnosed, and mean that patients are not worried, and help for hospitals. That helps them meet waiting list targets. If we can improve targeting of cancer, though, we can find it and hopefully find it earlier so that the outcomes are better. And lastly, by producing better reports, we can streamline communications, and all of this helps reduce costs. That's enough about Pi, our software, although we can come back to that in the discussion. I wanted to talk, though, as well about where we see this software going next. So right now in the UK, in England, about 56% of men with prostate cancer at first diagnosis, it's diagnosed as what we call late, so it's already advanced or metastatic. And for those men, treatment options are actually quite limited. 16% a couple of years ago, 12% in the most recent figures were diagnosed at first presentation with metastatic disease, and that's really limiting. The only way we can improve outcomes for those patients and really kind of help the treatment options for them is by finding the disease when it's much earlier, before it develops in the way that's been described earlier in this webinar. And for that, we need to do screening. Prostate cancer screening has historically been controversial because everybody used to get a biopsy. There was a huge amount of insignificant cancer found and a lot of unnecessary treatment, leaving large numbers of people incontinent or impotent for the rest of their lives. We're now working towards using our artificial intelligence, along with MRI and a number of other tests, to really make prostate cancer screening much more accurate. So there have been two really significant studies recently. The Prostagram trial run by Imperial College took individuals aged 40 plus with no prostate cancer symptoms and found significant cancer in 3% of them using MRI. In that case, for every cancer that was found, 10 biopsies were performed, which is actually quite a similar number to the proportion of biopsies for breast cancer. About 1% of women undergoing breast cancer screening actually have the disease, but about 10% receive biopsies. Another study called the Stockholm 3 trial has also recently been published. Here, there's a much more complex set of tests, multiple blood tests, as well as MRI. They also found about 3% of the patients who started the program had significant cancer, but had a better rate of biopsies. What we're hoping we can do with AI is reduce those numbers of biopsies further, and so really only find the disease that we need to find when it needs to be found, and actually make the process hopefully a lot cheaper and more accurate. And lastly, on personalized cancer treatment, I think the work that Daniel Woodcock was alluding to is really exciting. There's a tool that's actually available right now called Predict Prostate, which has been developed in Cambridge. This is available for patients with newly diagnosed cancer that isn't metastatic, and really gives them a great way of understanding the treatment options and the benefits and risks of each. And it's very interesting because in some cases, patients will choose one treatment option that maybe doesn't give them the maximum life expectation, but minimizes the side effects and really maximizes the quality of life. And this is now being used quite powerfully in practice. In the coming years, we will see all of the technologies that we've been talking about in the 4 talks so far today coming together so that we can understand disease as well as possible with as little information as possible, and do the fewest number of tests to rule out individuals who are healthy, but at the same time focus on finding cancer as early as we can, and then making sure that we've got the best information for personalized treatment. And so really, to summarize, we see AI as helping doctors deliver the very best diagnosis and treatment throughout a patient's life, diagnosing significant cancer early so it can be treated well, not treating when it's unnecessary, and treating just for as long as needed, and using in the best way possible to maximize a patient's outcomes and quality of life. Thank you very much. David, I think back to you.
Thank you, Dr. Rix, and thank you to all four speakers today for your presentations. I'm going to jump straight into questions. Conscious that we have run over a little bit, so I apologize about that. But I think the first there were a few questions that came in, and this is probably directed to you, Professor Wedge, really around kind of conceptually around sort of AI and machine learning, and I'm going to try and group a couple together. As one gentleman was asking, why do we actually insist on using this term AI when, you know, when we're not really actually at this point, you know, perhaps the strong AI that we're talking about that really has this kind of conscious mind, you know, in fact, it doesn't really exist at the moment. So is it a bit of a misnomer there?
Yeah. So I think Antony Rix alluded to this, that to some extent this is kind of governed by fashion. So sometimes it has been fashionable to talk about AI, and then other times it's been less fashionable, which is unfortunate, I think, because then we kind of get to arguing about terminology, which isn't really very helpful. But I think AI does represent a different way of working with computers. So the kind of traditional way of working with computers is that you know, programmers will write an algorithm which is a stepwise algorithm. So it carries out the first step, then the second step, and it's quite clearly defined. And the programmer decides the steps to be taken and what order they'll be taken in. With machine learning or AI, if you want to use that term, that isn't quite how it works. So the computer programmer will write the overall program, but it's not written in a kind of stepwise way like that. So most machine learning methods actually work in parallel. So they do lots of different processing in parallel rather than just going down one single kind of route to achieve the desired kind of outcome. And there's also this kind of idea of emergent properties. So the programmer doesn't decide what will be the output from the algorithm, and doesn't decide how it's going to do it. So what you often find is when you set off your machine learning algorithm, it actually comes up with an unexpected finding. So it might be using like a feature from an image to diagnose cancer, which is not you know, not a feature that any human would have looked at, but actually does achieve the desired objective, that it does identify, you know, the images that contain cancer. And that's, I think, one of the kind of key features of AI, that it can actually come up with unexpected results which weren't actually input by the computer programmer.
Fantastic. Thank you very much for that. And this, I believe, probably for you, Professor Brewer. In terms of the three of yours project, do you have any sort of timescale targets? I guess, you know, firstly for, you know, how long your project's running for, and then, you know, if things go according to plan, you know, touch wood, what sort of timescales do you think we might be looking at where we start to get this into the clinic and be talking about sort of personalized sort of treatment and making those sort of changes? What would be an ideal framework for that?
Yeah, I mean, it's a tricky question, because I'm sure many of you know that these things take quite a long time to get ratified in the clinic. But I mean, I would hope that we've got another 2 years left on this project, and then a validation study would need to be taken place after that, which is probably 2 years. And then after that, then we would be starting to think about testing it actually in the clinic after that. So that would be the starting point. But it all depends on how good it is and how the clinical community take to it as well. But these things can take quite a long time. But I would be hoping we're talking for like 5 to 10 years, that type of time frame.
Sure. Thank you for that. And Dr. Rix, this one's for you, and I think this probably touches on you know, the issue that Dr. Woodcock talked about in terms of the sort of black box and trust, or, you know, a few questions that came in around you know, how we sort of look at trusting the machines. accuracy and specificity of pi, you know, what does that look like, or sort of how do you assess that, and how can we make sure that people can trust, you know, that the machine is not going to be telling the, you know, radiologist something, or sort of taking over making those decisions?
So really good questions. Firstly, there's always a trade-off between sensitivity and specificity, and for different patients at different stages, they may have different preferences. But to give you an idea, the typical way we see pi being operated, it's more than 90% sensitive and about 50% specific. So its main benefit will be to help rule out biopsy for about half of patients who genuinely don't have any disease, and for everyone else it gives targets that will maximize the potential of biopsy. But exactly how that's used will depend on clinicians. I guess the kind of, you know, next question, there is sort of this perception about AI as being a massive black box, but actually the way it works is quite well understood, and the outputs in our case are those segmentations which you can visualize and which are checked always by a radiologist, and the lesions which are also always checked. It's actually really easy to interpret, and we've focused on making a system that's both explainable, so that, you know, we can communicate well what's going on, but actually also has a clinician in the loop, and I see that as being essential for the coming years, until frankly we've got really good evidence that AI is good enough on its own. The last and biggest issue is actually that isn't necessarily the algorithms, it's that we need to really be sure that these work in the clinic, in the real world setting. There is certainly a good amount of evidence that AI-like computer algorithms can suffer from problems when, for example, they're used on a different type of scanner, or they may work well with one data set, but not with another. Where we will soon be publishing a systematic review where we've looked at the literature, and shockingly, of several hundred papers that have been published on AI in prostate cancer imaging, not much more than 10 actually followed good processes of having held out data from external centers to really verify properly that the performance is good. We're addressing that within Lucida Medical by running a series of clinical studies with NHS partner hospitals and also with hospitals around the world, where we're specifically setting out to gather data from as many different settings, scanners as possible, and then firstly training the software so that it will work well across this very wide range of real world settings, and secondly keeping data back rigorously so that we can validate performance and be confident that when the system is used in the clinic prospectively, it will deliver the right diagnoses. And whilst we're not there yet, you know, we are confident that putting these two technologies together really does allow us to, you know, that will allow us to drive these accuracy levels up and then start to talk meaningfully about using AI to help unlock screening.
Thank you for that. And you mentioned, Dr. Rix, as a sort of follow-up, you mentioned you were starting sort of pilots. What are these next steps for Lucida and for Pi? What's the sort of?
So we're now taking baby steps to get the software being used in the clinic. Our first step is to work with a prospective adopter hospital to take a small number of patient scans, anonymize them, run them through the software, and then check that it's working correctly. Once we've done that check, we'll then support the clinicians as they start to use the software to make these decisions about whether patients should have biopsies or where the biopsies will be done. And we'll be continuing to monitor that, as we have to as part of medical device regulation, so that we can track over time that the software is actually helping deliver high levels of cancer detection and also the improvements in biopsies that we're targeting.
Thank you. And this question for you, I think, Professor Brewer. So you talked about when you were describing your tests, the way that you'd sort of mashed up the biopsies, and in doing so, it proved actually difficult, unlike in breast cancer, to then sort of extract usable information from that. And then once you've mashed the biopsies, you're then sort of unmashing them using the computer and AI. Is that correct, or am I explaining that? Because the question really came in was, so if you're mashing and then unmashing, what was the purpose of mashing in the first place? And that might be an oversimplification, but just trying to understand that process a little.
Okay, so I'll take this in two ways. I mean, firstly, you need a certain amount of RNA to perform these measurements, and so so that's why you mash up the cells, is so that you get enough material so that you can actually measure what's going on. But I mean, there is new technology that's currently coming into play called single cell sequencing. So that is actually sequencing a single cell. Okay, so some of the work we're doing at the moment on descent cancer is looking at some single cell data to see if it's doing, if the descent process is doing what we expect it to do. So that's on the horizon. Yeah, but basically, due to the constraints of the way we do these measurements, we are forced to, at the moment, take the whole, well, a part of the biopsy, mash it up to get enough material so that we can do these measurements.
Brilliant, thank you for that. And conscious of the time, we are now at our time. So I just want to thank everyone for coming today and for all your questions. I know we didn't get through all of them, but I hope we were able to answer most of them satisfactorily. And thank you, of course, to all four speakers for a really interesting set of presentations. And I hope we will be able to have you all back at some point in the future, maybe to give us some updates on where things are going for each of you respectively. So thank you all, and please, to everyone who's attended, I know there's many of you know there's a post-webinar survey. Also, that includes this question asking for any thoughts on what you'd like to be included in next year's webinar series. So all suggestions are very welcome, because they will help us plan next year's series and how we can support you better. Thank you all again, and wishing you all a wonderful day.